Okay, so this video is about statistical analysis, and you might be saying, "Oh no, I didn't think I signed up for a math class." Um, but statistics are very, very important in science, and really, it's only through statistical testing that we can be confident in the fact that we have um, disproven an idea or not. And so that's really important to understand here. And what we need to do is break down some of what you think statistics is and what you may not know yet. Hopefully you do know some of these terms that we're going to talk about and then apply. So what I've done here is written out your students will be able to objectives. I'll go through these, but I just do want to point out that there are no corresponding pages in your book because this is not required curriculum for your tests per se. It is absolutely required for your internal assessment and science fair, which means that it is important to cover and will contribute to 20% of your IB grade. So let's go ahead and talk a little bit more directly about this. So what you need to be able to do is distinguish between descriptive statistics and hypothesis testing. And you might know a little bit more about descriptive statistics than you might realize, but hypothesis testing, my guess is that you may not know that well. The second one is to define standard deviation, normal distributions, and error bars. Third is to explain how standard deviation is useful for comparing the means and spread of data between two or more samples. And four, calculate the means and standard deviations of data sets and graphically representing those values. And those are good. We're going to do that with Excel or perhaps Google Spreadsheets if you don't have Excel at home. But Excel is going to be the tool that we want to use the most, unless you know how to use Logger Pro in more detail. And the last one for this video is to explain how correlation does not mean causation, and to ensure that you know how to recognize correlation when you read it and you see it, and understand that that doesn't necessarily mean causation. Okay, so let's go ahead and jump into this. So let's talk a little bit about what descriptive statistics and hypothesis testing are. So I'm going to scroll down a little bit here. So when we talk about descriptive statistics, what I mean by that, and what statisticians mean by that, are those terms that you are familiar with, like mean, median, and mode. And I won't define those for you because I think you know what those are, and if you don't, look them up. But there's another term here that you may want to become familiar with if you're not already, which is standard deviation. And standard deviation is an important term to make sure you understand because it does describe something about your data, and it's a measure of the spread of your data, of your data points around the mean or the average, right? So high standard deviations mean that the data is more spread out, and less standard deviation means that it's not as spread out. And so one way you can think about that is if you had like you were playing darts and you had a target, and you hit, let's say you hit a bunch of times right around this spot right here, as opposed to hitting all over the board, maybe even a bullseye there, but hitting all around the board. These hits are far more spread out, which would indicate a higher standard deviation or a higher spread of the data around the average, whereas these are more closely clustered, which would make a smaller standard deviation or lower spread of the data. And really the key that we want to look at for reliability and, well, really for reliability would be a low standard deviation. So that's going to be the key here, despite the fact that you might have even hit the target right on with, but because there's a wide spread, it's less meaningful because it's less predictable that you're going to get meaningful data or meaningful points. Okay? But the key here is really that it's not even all that meaningful because this is just describing things. It's not really comparing two groups of data. So there's another idea called hypothesis testing, and that's key here. So you know what a hypothesis is, I hope. I hope you've been taught that it's not an educated guess, but rather a prediction. And it does not need to be written in an if-then sentence. It's just basically what you think is going to happen and why your groups are different, right? So in hypothesis testing, there are two main hypotheses you need to make sure you worry about. One is the alternative hypothesis, which is specific about it in regard of, it's specific regarding which group you think will outperform others, but making sure that it's operational, or in other words, it's testable. In other words, it clearly indicates how the data will be measured, both in the independent variable and the dependent variable. But there's another idea, which is the null hypothesis. That means basically that there will be no difference, and I'm just writing the shorthand, that there will be no difference in the data, meaning that no matter what levels you change the independent variable within your experiment, there will be no change in the dependent variable or the data. And really the idea here with hypothesis testing is that the attempt here is to refute the null hypothesis. If you refute the null hypothesis, what you end up doing is that you're supporting your alternative hypothesis. If you cannot refute your null hypothesis, that means that there is no difference. Oops, I didn't mean to cross it out. There is no difference between your independent variable. But that does not mean, let me make this caveat, that does not mean that differing the amount of your independent variable makes no difference. It means that within the range of your changes of your independent variable, there was no difference that you found according to the dependent variable or variables that you used to collect data. So you need to understand the scope of what hypothesis testing is. And there are a few tests that we're going to learn about this year. And I'll tell you when we're going to learn them. But very immediately we're going to start learning about something called a t-test, which is basically when we compare two groups or two levels of independent variable, or an ANOVA test, which stands for analysis of variance, which is more than two groups. We're also going to learn about chi-square testing. Chi-square testing when we get to genetics. And we'll also talk about some Pearson R correlation stuff. Okay, which leads into the correlation stuff that I'm going to talk about a little later. So these are the two that we're going to talk about. I'm going to show you how to do these in Excel now. When we get to genetics, we'll talk about chi-square stuff. Okay. Let's go back to the objectives list. All right, so what we've done, I hope, is been able to distinguish between descriptive statistics and hypothesis testing. And we have defined standard deviation, but we need to talk a little bit about normal distributions and error bars. So normal distributions are maybe what you've known as a bell curve. So if we had a graph and I'm going to put some measure of frequency on the y-axis, and let's say, I don't know, to do some hypothetical test scores, and we were to compare kindergartners to juniors, and we had some kind of marks here for the different gradations. If our average score for kindergartners is represented, let's say right here, and our average score for 12th graders, let's say, or sorry, for juniors is here. So kindergartners have a higher score. Let me make a little key here for that. So this is preschoolers, or I don't know if I said preschoolers or kindergartners. Either way, young kids. And this purple line is going to be for juniors. So your age kids. Okay. So those are the averages, and so here would probably be 0, right? And this is the max score. And I would ask you, which group of kids did better? You might say, yeah, let me ask that. Which group do you think did better? And that's something you've been trained to look at is just the average, but you really don't know the spread of the data. There might be some preschoolers. Remember, this is an average, so their outliers can really impact this. You might have one preschooler who did really, really awesome on this test, and one junior who did really, really poorly on the test, and that skews the averages, and so that's not giving you a holistic picture of the group. And so what we need to do is understand what a normal distribution is. So I'm going to use my purple here. So let's say that our average score has a frequency, a pretty high frequency, but what we would typically expect in a normal distribution, right? I'm going to write that up here. What's called a normal distribution is that from that data, from that mean, we would predict some kind of bell curve like that. Well, this should curl a little more like that. Right, so there's a little bit of a bell curve there, and we would predict a similar bell curve, and maybe there's a higher frequency of this average score here, right? So we might have something like this. I don't know. I think you understand the idea. Actually, it would probably be a little skinnier, a little tighter in, but either way, here's the average, and here's the average, right? So here are, put an X there. So we still see that the averages are different, but what the bell curve does is shows us the distribution of the scores. And so there's going to be some value that we can calculate, a standard deviation, that allows us, and that should be directly above it, and I apologize it's not, there'll be some value from which that we can go minus and plus and draw these lines down to represent plus or minus 1 standard deviation. And that really typically represents about 68% of the data. So about 2/3 of all scores fall within that range. And so when we have a higher value to go up and down from the mean, a bigger value means a larger spread for that 68%, and a smaller value means that those 68% of the scores were really tightly held around the mean. If we go another standard deviation, then, and remember, let's say that standard deviation is calculated at this, let's say that this average score, let me use some real numbers here. So let's say that's a 70%, and this is a 50%, right? So let's say that the standard deviation is plus or minus 10, right? So this line right here would be at 80 and 60. So 68% of the scores fell between a 60 and 80%, and then another 10% up represents, well, and another 10% down represents 95 total, 95%, or 95% of the scores. And yet a third, that's 2 standard deviations, and a third one would represent 99% of the scores, right? So on both sides, total, everything in the middle, right? So this is how we think of a normal distribution, that scores would fall out in nicely distributed terms, and really that only happens when you have a really high sample size or lots of data points. But the same would be true for this, for the junior bell curve. And what we need to look at is the spread of data around the plus or minus 1 standard deviation. So if for the juniors the standard deviation happened to fall to the right of this -1 standard deviation line for the preschoolers, then we would say that there's overlap and that there's not likely a difference. Even though the means are different values, the spread of data is so wide that we can't, with confidence, with a 68% confidence level, say that there's a significant difference. And you might say 68%, that's not very precise. That's not very confident. You're right. 68% is not our norm. In biology, we go with a 95% confidence level. So we'd have to look at plus or minus 2 standard deviations. And if our plus or minus 2 standard deviation line is right here, which is really close to our mean, we can probably guess that plus or minus 2 standard deviations on either direction from the mean is going to overlap with the range for the preschoolers, which would indicate that there's not likely a difference between these two groups. But if the standard deviations are really small, which can happen with more data, because any outliers get canceled out, you might just see that these groups are in fact different. Okay, so this is one way to represent that. Let me show you a different way to represent it, which I don't expect graphs like this. You certainly can, but you could also have a graph that looks like this. So here we would have, so because these are two distinct groups, preschoolers and juniors, what we would have maybe is a P for preschoolers and a J for juniors. And over here on the y-axis, we would have scores, right? So we're not accounting for frequency anymore. But we have, I said a 70%, so it might be a bar graph because we have different categories. It's not continuous data, which might present another problem for data sets, and what kind of data you should be trying to collect. And for juniors, we said it was about 50%, so not as high, right? And again, the top part is just representing the average. And sometimes this bar graph can be misleading because it represents data within it, not that point. And then what we would do is add error bars. So if this is my average value, what we would need to do is if this standard deviation for the preschoolers is 10, I would go plus or minus 10. It's hard to see that white. Plus or minus 10 over that bar. And let's say the juniors had a 15 point standard deviation, they would have a wider, I'm going to over-exaggerate, a wider error bar, right? So those error bars on a graph can represent one of two things. They can either represent standard deviation or they can represent the range of the data. Right? So the high and low point. But what we're going to use in our testing is standard deviations. And so when we look at this data and you see that there is a wide overlap, so if we were to basically push these error bars all the way left or overlap them, we would see that those error bars would sit on top of each other, which would mean that there's not likely a difference. But this is all descriptive statistics. This is not hypothesis testing, which leads us into the next part where we need to understand what a t-test would do. Because we have two groups here, a t-test is what's most appropriate. Okay, so what we've done here is explain how the standard deviation is useful. I hope that you understand that. By comparing the means and spread of data between two or more samples. We've also looked at what a normal distribution is and looked at what error bars are and what they try to communicate. So I'm going to go to objective 4 in just a second, but let's go to number 5 first. Okay, so we're going to talk about correlation and causation. Okay, so correlation and causation. So correlation basically just is indicating trends and patterns. And so if we were to have a graph and we saw the data points follow in this line, you might think that as the x-axis increases, the y-axis increases too. And this is what, right? This is called a positive correlation, right? And because it's all pretty much around this line, it's a pretty strong correlation. And we would need actually a Pearson R correlation value or an R squared value to look at that. But there's a very important distinction here, is that unless the data on both axes was determined or calculated or measured through experimentation, we don't know that there's causation involved, right? So causation can only be determined, causation is determined by experimentation, okay? So only when you're conducting an experiment and changing only one variable do you know that if the data appears to be different through statistical testing, can causation be determined. If we were to look at, I'll give you a hypothetical here. So if we were to compare two data points that we just collected, like let's say ice cream sales and shark attacks, right? So I think that you would probably expect to have a positive correlation. As there are more ice cream sales, there are more shark attacks. But the problem here is that they may not be related and they may not be causing one another. It's probably the case, right? Because someone buys ice cream, that they're more likely to get attacked by a shark doesn't make sense. Or being attacked by a shark causes you to go and buy more ice cream doesn't make sense, right? So you don't know which variable is causing the other. So there's a problem there, but you also don't know if there's another variable altogether that's causing this correlation. So this positive correlation is more likely, just out of using common sense, caused by the fact that ice cream sales go up during the summer, which is when people also spend more time in the ocean, which is where the sharks are, right? So there could be a third variable altogether that's causing it. So correlation is really important to make sure you understand. And we see this all the time reported in the news. Sometimes people will look at medical data and say that having a glass of wine helps with heart disease. And what they're looking at is they could be looking at two different things. One thing that they could be looking at is a correlational study. Let's look at the people who drink wine regularly and look at their heart health. Well, the problem with that is that the people who can afford wine every day probably, or may live healthier lives, have access to maybe different kinds of food, have health insurance, have a different kind of economic status, which can definitely play into health. So that's not a causative study. That's not an experiment, and you can't make that claim. The alternative that some people actually do claim based on experimentation is that there's a chemical in wine that when dripped on cancerous cells in a petri dish in a lab can deter cancerous growth. They are making the claim that drinking wine helps with cancer, right? So one chemical causing that is fine, or helping prevent that is fine, but when you add other chemicals that are in wine and other variables, like it's not a single cell or tissue, but when you apply it to a body, there are all other kinds of variables at play, and so that's not logical. That's not a logical extension of that data. A lot more has to be done and tested to get there, okay? So I'm looking at my time. I'm at 23 minutes. I'm going to pause it there and make a separate video about Excel to show you how Excel works, okay? You're going to see a series of questions now based on all the content in this video. These are IB questions. Do your best to answer them. Anything that you get confused about, we will certainly talk about if you bring it up in class.